All cells go through cell cycles. In cell cycles, the cells grow, maintain, and reproduce in order for the organism to perform various functions of life. In this video, we will demonstrate the cell cycle by using Play-Doh models, along with images of actual human cells in different stages of the process. Three main phases of the cell cycle are interphase, mitosis, and cytokinesis. In interphase, the cell grows and prepares for mitosis. In mitosis, the replicated chromosomes are separated into two nuclei. And finally, in cytokinesis, the two nuclei, each containing a set of chromosomes, are separated into two cells, completing the division of the cell.
The first stage of mitosis is prophase. First, the ribosomes make the nucleolus. A part of the nucleus disappears. After, the nuclear membrane breaks down and releases the chromosomes. Then, the chromatins condense and become 1,000 more times compact than they were in interphase. The chromatin condenses into two rod-shaped structures which are called chromosomes. This is called chromatin condensation. In the process, microtubules also replicates, and the two copies move towards the two poles of the cell. Next, the mitotic spindle continues to grow, and some microtubules start to capture chromosomes. Microtubules combine to chromosomes at the connecticore, which is a patch of proteins found on the centromeres of each sister chromatid. Sister chromatids are two identical copies formed by the replication of a single chromosome, and both copies are joined together by a common centromere, which is a region of DNA where the sister chromatids are tightly connected. In metaphase, the mitotic spindle has captured all the chromosomes and lined them straight up at the metaphase plane and the equator of the cell. The metaphase plane is an imaginary plane perpendicular to the spindle fibers of a dividing cell, and the equator of the cell is the central plane in a dividing cell. This alignment occurs with the help of the motor protein called the centromere-associated protein E and another protein called dynein.
During anaphase, the sister chromatids begin to separate. The spindles attached to each sister chromatid reach from the microtubulars on opposite poles of the cell. They begin to contract back to the poles and pull the sister chromatids away from their partner chromatid. As the spindle retracts, it causes the chromatid to bend and form a V shape as it pulls away. The centromere that holds the sister chromatids together at the equator of the cell divides as the sister chromatids are pulled apart. The spindle fibers begin to disintegrate after the chromatids are pulled away from the centromere and their partner chromatid. Telophase is the final stage of mitosis. During telophase, the chromatids are located on opposite ends of the cell. Now that there are two identical sets of chromatids on each side of the cell, a nuclear membrane develops around each set. While the nuclear membrane forms, the nucleolus reforms as well as the ribosomes. These two structures are both formed within the nuclear envelope.
At the end of mitosis, cytokinesis, the division of cytoplasms and organelles to form two genetically identical daughter cells, takes place. After cytokinesis, each daughter cell enters the interphase once again and continues the cell cycle. Mitosis and cytokinesis have a direct connection to the treatment of cancer. Unlike normal cells that replicate and die, cancer cells are frequently immortal, since the mitosis of cancer cells cannot be regulated, causing them to replicate indefinitely. Nowadays, patients with cancer often receive chemotherapy treatment such as Taxol. Taxol interferes with the spindle checkpoint that ensures the chromosome pairs to align correctly before the cells divide. If this process is interfered, cell suicide will occur. Taxol inhibits the spindle checkpoint of all rapidly dividing cells from working properly, leading to the death of tumor cells. In this way, cancer cells are killed before they enter the cell cycle once again, thus helping treat cancer in an early stage. If Henrietta received such treatment, it is possible that her cervix cancer could be cured. However, she didn't get the now common treatment, causing her cancer cells to replicate and tumors to proliferate continuously.